Yeah.
Let me start recording. Okay, so welcome everyone. And today in our usual Knowledge Portal Network focus group time slot, we have a guest speaker, Dr. Paul Titchenell. And Paul is an associate professor of physiology at the University of Pennsylvania, which is my undergraduate alma mater. So I like your picture, Paul. I can see things recognized there. And Paul is our colleague in the AMP-CMD consortium. He did his undergraduate work at Dickinson College and his graduate work in physiology at Penn State, where he investigated the role of VEGF signaling in diabetic vascular complications. He went on to do a postdoc in Morris Birnbaum's lab at the University of Pennsylvania and then stayed there in the Department of Physiology as a professor. And his current work focuses on metabolic regulation by hormones and nutrients, and I think we'll be hearing more about that today. So Paul has said that if you have a question, you can interrupt him during the talk. He's happy to have this be interactive, and you can just type your questions into the chat box, or actually you can use the Zoom function to raise your hand and actually speak. And this is a focus group format, so it's not like a webinar where you're all, where no one else can speak. Actually, the attendees can speak here if they want to. So I think that's it. And welcome, Paul. We're looking forward to hearing your talk.
Great. Thank you, Maria. I appreciate the introduction. And it's great to hear that you also enjoyed Philadelphia. And I think you'll note that the picture on the left was before the new hospital was built. So that was the skyline view from my office prior to the end of 2022. But the new hospital on the right is the stark contrast. But
okay, yeah, I mean, I was there a very long time ago, so.
No problem. The skyline has changed a little bit, but we're there. It's still a great place to do science. So, yeah, thank you so much. I'm looking forward to sharing with the group some of our recent work investigating the role of insulin action and insulin signaling, specifically focusing on the serine threonine kinase AKT in the control of muscle growth and metabolism. And as Maria mentioned, I'm happy to make this interactive. So if you post questions throughout the talk, I'll do my best to answer them. Okay, so to get started, my lab is quite interested in trying to understand the molecular mechanisms that underlie insulin resistance. And the way I want to define insulin resistance is the way that it was originally defined by Root in the late 1920s, where he noted that some individuals with diabetes or hyperglycemia required very large and significant amounts of insulin to normalize blood sugar. And he termed that insulin resistance, and says that seems to be justified based on the amount of insulin that were given. And it was further really refined by Himsworth about a decade later, when they did a classic experiment we do in the diabetes field, is you do a glucose insulin tolerance test. So you had individuals that were all considered to have frank diabetes, so they were hyperglycemic at baseline. And he did a simple experiment where you inject glucose and insulin into those individuals, and you have a subset of them here marked with the Roman numeral 2 that are insulin sensitive, meaning that if you gave insulin plus glucose, you can see this nice drop in blood glucose levels that are relatively constant over time. The x-axis is time. However, there were other diabetic individuals that if you do the same exact experiment, they were seemingly insensitive to insulin, and that if you gave glucose plus insulin, their blood sugar rose over time. And so here is really the basis of the difference between an insulin sensitive individual and insulin resistant individual. Obviously, these are both diabetics, and we now know that these insulin-sensitive individuals are more commonly referred to now as type 1 diabetics, and these insulin-resistant individuals are now commonly referred to as type 2 diabetics. And if you look a little bit closer at the molecular basis of this insulin resistance, you can see that there's a significant impairment in insulin's ability to control peripheral glucose disposal. So this is a study in the early 80s by Kolderman et al., doing hyperinsulinemic euglycemic clamps coupled with tracers to measure the rate of glucose disposal throughout the body. And here on the x-axis, you just titrating different insulin concentrations, and the y-axis is reflecting the glucose disposal rate. Controls are in the closed circle. So if you give increasing concentrations of insulin during a clamp condition, you can see you can dose dependently increase glucose disposal. That makes a lot of sense. Insulin drives glucose into tissues. However, in individuals with type 2 diabetes, either with obesity or non-obese individuals, you can see a shift to the right of the dose response curve. And this is what I would like to define insulin resistance in this talk. So there's a shift to the right. So meaning you need more insulin in an insulin resistant individuals to have the same degree of glucose disposal that you would in the control. So for this case, let's just take the example. If it's 100 in a control to get this rate of glucose disposal, you need roughly 5 times the amount of that in an insulin resistant individuals. That's the definition I would like to use. If you look closer at about what tissue makes up this this contribution, we know muscle, for the majority of us, should be the largest sink for glucose in the body. And if you look closer using forearm biopsies during clamps to measure specific glucose uptake into skeletal muscle, you can see that consistent with this defect in glucose disposal, you can see in obesity and in insulin resistance, you can see a significant decrease in whole body glucose disposal that is very much correlative to a decrease in glucose uptake, specifically in the skeletal muscle. So we've known now for 30, 40, maybe 50 years that type 2 diabetics are insulin resistant, and they clearly have impaired muscle glucose uptake. Okay, so here's just more of a conceptual cartoon about from a series, multiple decades of work, really elucidating several critical steps in this process. A lot of it was pioneered through the discovery of the glucose insulin responsive glucose transporter here called GLUT4. And we know that insulin can bind through its receptor tyrosine kinase, cause a signal transduction event to drive GLUT4 to the plasma membrane, and then thus increasing glycogen synthesis and glucose oxidation. And what I've just shown you is that in insulin resistance, there's a failure of glucose uptake. And in a series of studies led by many pioneers in the field, have documented that there's a defect in the ability of GLUT4 to translocate to the plasma membrane during insulin resistance, and that's associated with this decrease in glucose uptake. Okay, so a big question in the field, and what we're going to talk a little bit about today, is where is the biochemical block in GLUT4 translocation and glucose uptake? Is it proximal to GLUT4? Is there a failure of insulin to get to the signal to GLUT4, or is it distal? Is there some part of the cytoskeleton or the trafficking events that are impaired to that thus prevents GLUT4 getting to the membrane? Okay, we're going to explore really these proximal mechanisms and test the hypothesis, exploring the role of AKT in mediating GLUT4 translocation. So here is a canonical insulin signaling pathway. Insulin through the insulin receptor, which is a receptor tyrosine kinase, leads to the recruitment and activation of IRS proteins and subsequent activation of the canonical PI3 kinase AKT pathway. And AKT is a serine threonine kinase that is involved in a host of metabolic responses. Work by Lori Goodyear in the mid-90s showed in obese insulin resistant individuals that insulin fails to phosphorylate insulin receptor substrates, IRS substrates or IR, so suggesting that there was impairment here. Subsequent studies looked even further downstream at PI3 kinase, again, using radiolabeled assays in an obese insulin resistant individuals. You can see a decrease in PIP3 generation. And then later work has shown using immunoprecipitation of AKT followed by in vitro kinase assays looking at phosphorylation of GSK peptide, a target of AKT, and has demonstrated that in type 2 diabetics, there's a reduced AKT activity, at least on the concentrations of high insulin administration. So these are all consistent with signaling defects throughout the proximal insulin signaling cascade. But a major question remains, are these defects simply a consequence of insulin resistance, which typically happens with signaling pathways? They become downregulated if the ligand is increased, or are they causal, right? Are there specific inherent changes in these pathways that can then drive insulin resistance? Okay. And some clues started to come in the early 2000s by Maury Birnbaum's group, where they generated whole body AKT knockout animals. In particular, one animal, the AKT2 isoform, which is most abundant in metabolic tissues like liver, muscle, and adipose tissue. And they demonstrated that this mouse model, just by changing AKT2, by knocking it out in the whole body, was sufficient to cause insulin resistance. Okay, so here's a clamp study, again, measuring total glucose disposal. And you can see in the knockout animal a significant reduction in total glucose disposal, i.e. insulin resistance. And this was associated with impairments in skeletal muscle glucose uptake, at least in the EDL at sub-maximum concentrations, okay, suggesting that AKT2 is involved in glucose uptake. Further validation of the role of AKT and the causal relationship to insulin resistance was really put forward by Stephen O'Reilly and his colleagues, where they identified, these are quite rare mutations, but a family with severe insulin resistance that had mutations in AKT2. And this is the proband here. And you can see this is just a clamp insulin infusion rate, or I'm sorry, glucose infusion rate. And you can see this individual has very, very high insulin levels, yet the infusion rate is very, very low. Again, higher infusion rate would be more insulin sensitive. Infusion rate here is very low. These individuals are very insulin resistant. Okay, so collectively, there's both mouse and human data to suggest an important role for AKT in mediating insulin action and glucose homeostasis. So as a result, for 20 years or so, there's been a popular model that has been put forth to really try to connect the dots between obesity, diabetes, and insulin resistance through through exploration of signaling events involved in GLUT4. And here's a classic study that people will do in OB mice, where you do titration of insulin. You can see a nice robust increase in glucose uptake in skeletal muscle that is impaired in OB insulin-resistant mice, right? So at various concentrations, you see a failure to stimulate glucose uptake. And this is just data from our lab, just doing a simple Western blot analysis, looking for the activation of AKT. Here's monitored by phosphorylation status. You can see insulin leads to robust activation of AKT at the supraphysiological dose. And then in OB under these conditions, there's a defect in AKT phosphorylation. Okay, so putting these two together, a common hypothesis is that there's a defect in AKT phosphorylation and activation that is thus driving this insulin-resistant phenotype. So putting it into context, AKT lies downstream of the insulin receptor, and somehow AKT is involved in GLUT4 to facilitate glucose uptake in muscle, right? And work by Jerry Schulman's has really put forward the idea of ceramides being a link between obesity and fatty acids, work by Scott Summers and others, Chris Newgard for branched-chain amino acids, have all really implicated changes in lipid species that have impinged upon proximal insulin signaling to reduce AKT and thus, in turn, cause insulin resistance by blocking glucose uptake. Right. So this is sort of this popular model I'll put forth that underlies insulin-resistant phenotype. However, I would argue that it's never been directly demonstrated that a reduction in AKT activity alone was either necessary or insufficient to cause insulin resistance, okay? And so for this question, to really test this popular model, we set forward to make mouse models that specifically lack various AKT isoforms in the muscle to really test the specific role of AKT in glucose metabolism and muscle growth. And that's what I'm going to be focusing on for the majority of the presentation now. Okay. So what a talented postdoc in my lab has done, Natasha Jaswal, she generated various AKT-specific skeletal muscle knockouts, either taking advantage of the AKT2 flox allele or doing the combination of AKT1 and AKT2 to get rid of both AKT isoforms in muscle. She also did AKT1, which we're really not going to focus on because it only makes up roughly about 10% of the AKT that's expressed in muscle. Okay, here you can see the deletion of AKT2 and the significant reduction of the pan-AKT signal. The rest of the signal is really coming from non-myoblast cells and a whole muscle prep. The specific cree that we can use is the human skeletal actin, which affords skeletal muscle specificity. So here's just taking a double knockout of AKT1 and 2, and you can see the skeletal muscle, it's lost, but it does not affect liver, and importantly, the heart. So it's cardiac excluded, nor does it delete in the white adipose tissue. AKT3 is the third isoform of the AKT family. It is typically not expressed in skeletal muscle, nor is it upregulated in the absence of the other two isoforms. And here's just a positive control showing you that it's very much abundant in the brain. Okay, and then lastly, just for, again, another control, if you knock out both AKT isoforms and you stimulate that animal with insulin, you're still able to activate phosphorylation of the insulin receptor. So this is really indeed affecting downstream at the level of AKT and not affecting proximal insulin signaling through the insulin receptor. So we're really isolating the effects of AKT in the control of muscle biology with this approach. Okay, the first thing you can do typically, obviously, from a skeletal muscle perturbation is you can first just weigh the muscles. And the first thing that was kind of surprising to us was that the AKT2 knockout really had minimal, if any, effects on muscle weight. And this is thought to make up roughly 70–80% of the AKT that's expressed in muscle. All right. If you look at both the double knockout, AKT1 and 2, you can see significant reduction in the muscle weight of these animals. Okay, so there's about 30–40% reduction in the muscle size. And we're going to focus more on that in the second half of this talk. But I want to focus here initially on really the glucose and insulin phenotypes. Okay, so again, AKT2, whole body knockouts were insulin resistant. However, if it does not affect muscle weight. The question is, does it affect insulin action? So we can do a canonical Western blot analysis where you just do insulin treatment with control, and you can see nice activation of AKT and its downstream targets, including mTORC1 as determined by S6 and 4-ABP, and the direct AKT substrates PRAS40. If you delete AKT2 alone, again, you can get rid of roughly 80–90% of the AKT active phosphorylation, but downstream signaling of AKT was maintained. So this really tells you only need 5–10% of active AKT in skeletal muscle to transmit the signal, okay? If you get rid of both AKT isoforms, you can completely remove phosphorylation, or you can completely prevent activation of AKT. There's no AKT in these muscles, and you can completely prevent downstream signaling. Okay, so we have really two models. One is clearly reduced AKT phosphorylation, but normal signaling, and one is a model where there's really no AKT activation. Okay. When we did some initial preliminary glucose phenotyping, we were struck by the fact that AKT2 knockouts alone had no discernible phenotype at all in terms of glucose homeostasis, insulin sensitivity as measured by an insulin tolerance test. So the lines are superimposable on the control. What was even, frankly, more surprising to us, though, was that a muscle that had both AKT isoforms, the AKT double knockout, yes, and did indeed have an effect on muscle mass, but had relatively modest effects on glucose tolerance and insulin resistance. This was very surprising considering the hypothesis is that AKT serves as a key insulin responsive kinase that was involved in GLUT4 translocation. So we really wanted to investigate this much further, right? We did clamp studies, looking at insulin action in vivo. And if you normalize to the muscle mass, so if you do echo MRIs prior to the experiment and say, what is the infusion rate relative to the amount of muscle that this organism has, the insulin sensitivity readout was normal. So infusion rate was normal, and whole body glucose disposal in an AKT muscle knockout was normal. And this is quite surprising because these animals do not have AKT signaling in muscle. All right, but they do have a defect in muscle mass. Consistent with the whole body disposal rates, if you look at that tissue-specific glucose uptake during the clamp, you can see similar rates of glucose uptake in slow and fast twitch muscles and then mixed muscle, okay, all consistent. Really to verify these in vivo findings and see if they're cell autonomous or not, we can isolate tissue. This here is the soleus tissue, and measure glucose uptake ex vivo. So you can stimulate with insulin in a control. You can see this robust activation of glucose, or robust stimulation of glucose uptake that is suppressible by Ward-Manin, which is a PI3 kinase inhibitor. Okay. And again, consistent with the in vivo phenotypes, an AKT muscle knockout has normal rates of insulin stimulating glucose uptake that is indeed suppressible by PI3 kinase. So these data really support the concept that there is the existence of an AKT independent pathway in skeletal muscle to drive glucose uptake, but that insulin does indeed require PI3 kinase to signal. All right, this is glucose uptake, and we know GLUT4 tracks with glucose uptake, but just to kind of answer the question that GLUT4 was indeed translocating independently of AKT in these tissues, we developed, this is again by Natasha, developed an assay using single fiber isolation, taking advantage of a trick that David James really first put forward. Is if you have a GLUT4 construct that has a MIC tag inserted into the exofacial loop, you can do in a non-permeabilized manner, you can look at the amount of GLUT4 that gets inserted into the plasma membrane by looking at MIC expression. So we can infect single fibers and then with this construct to look at insulin-stimulated GLUT4 translocation. All right. And M-cherry is on the cytoplasmic tail, and that would be a good readout of just expression. This is what it looks like in a control system. If you, again, here's the MIC staining in an unstimulated condition. There's very, very little MIC expression. There's very little GLUT4 under these conditions at the plasma membrane. This muscle does have GLUT4, or I'm sorry, this fiber has GLUT4 expressed as indicated by the M-cherry. If you give insulin, you can see this robust increase in MIC label consistent with an increase in GLUT4 that's being inserted in the plasma membrane. If you do this experiment in a in an AKT double knockout, you can see a similar degree of GLUT4 that is now at the plasma membrane. So consistent with the glucose uptake biochemical assays we just showed you, here is directly measuring GLUT4, and there indeed GLUT4 and glucose uptake appear to be normal in the AKT knockout. Okay, so just kind of putting forward this in a more of a conceptual point of view, this is the traditional view, is that insulin signals through AKT and controls these RAB gaps. There's different flavors of these. I'm not really going to focus on that today, but that is involved in GLUT4 translocation to stimulate glucose uptake. Okay. What I think I've just shown you is the existence of AKT independent pathways. And this is really the first time that this has been demonstrated in an in vivo system where there has to be an additional pathway that exists that is typically suppressed by AKT to drive GLUT4 in response to insulin. And obviously, we really want to understand what that pathway is. But first, I just want to pause, and we did publish this finding a couple of years ago now, and we were very reassured the end of last year that the Katawaki group published very similar findings, suggesting that, again, AKT in skeletal muscle is dispensable for glucose uptake. So this is from the Katawaki group using a separate skeletal muscle specific Cree to generate muscle specific AKT double knockouts here. And you can see this is the insulin treatment. And similar to what Natasha published a couple of years ago now, these animals displayed normal rates of insulin-stimulated glucose uptake in either the soleus or the EDL. So we feel quite confident and reassured that this phenotype is indeed real and robust. Okay, so there is an existence of an AKT-independent pathway for glucose uptake. The first, I think, question we wanted to really ask, in giving that all of the previous data has really put forward AKT as being a critical kinase involved in insulin sensitivity, and a lot of work by Maury Birnbaum, David James, and others, especially in adipocyte models, we wanted to ask in vivo, is the same true? Is AKT dispensable in adipocytes to drive GLUT4 translocation and glucose uptake? So we generated AKT2 fat-specific knockouts, taking advantage of the adiponectin Cree, and you can see the reduction in AKT signaling in this model. But unlike skeletal muscle, when you knock out AKT2 in vivo, and specifically in fat cells, what Natasha has shown is that these animals, and these are primary adipocytes isolated from the fat-specific AKT2 knockout. You can see they are very resistant to insulin in terms of stimulating glucose uptake. So normally insulin drives glucose uptake in fat in primary adipocytes, and in primary adipocytes that lack AKT2 from the fat-specific AKT2 knockout, you can see a significant impairment. And this results in significant insulin resistance as determined by an insulin tolerance test. And we have clamp studies that suggest the same. So to put this all together for the first part, mice lacking AKT1 or 2, specifically in the skeletal muscle, surprisingly appear phenotypically normal. Okay. There's a tremendous amount of spare capacity that exists in the AKT signaling pathway that only 5 to 10%, based on our calculations, is required to transmit a downstream signal. Okay. And it doesn't appear, at least in vivo, that there's true substrate specificity for AKT2 or AKT1 in skeletal muscle. They both can functionally compensate for one another.
Paul, can I interrupt you?
Yeah, please.
This is Alan Addy.
Hi, Alan.
Can you go back a slide?
Sure.
So this is a very surprising result because you're seeing a huge effect on glucose clearance in the insulin tolerance test. And, adipose is not
Yeah.
supposed to be playing a big role.
Yeah.
So do you think this is an endocrine effect of adipose on muscle?
Yeah. So, I'll go further to say, actually, we know that from clamp studies, when you make a fat-specific AKT2 knockout, you actually cause profound systemic insulin resistance. So we see muscle insulin resistance in that model, and we see actually a complete failure of insulin to suppress hepatic glucose output in the fat-specific AKT2 knockout. Mechanistically, there's a lot of changes. Some of those include adipokines, some of the fan favorites of some, like adiponectin tracks. We see big changes in fatty acids, and meaning that insulin fails to suppress fatty acids in an AKT2 fat-specific knockout that maybe I would argue contributes to the liver phenotype. So the shorter answer is, I'm not trying to paint the picture that the fat-specific glucose uptake phenotype alone is driving the systemic change. It's leading to a series of secondary effects that are clearly contributing to an insulin-resistant phenotype. And frankly, to us, it looks very similar to what Mori published almost 20 years ago as the whole body knockout. We think a majority of that phenotype can be ascribed to to a defect in AKT signaling in fat that then causes these secondary effects. Hopefully that answers your question, Alan.
I don't want to keep you stopped here. We can come back to it later if you want.
Okay.
So I'll interrupt as long as we stopped here. We also have a question from Eve Fain about were the mice in conclusion 1 allowed to be active? So I guess that's your first bullet point here on this slide.
Yeah, they were allowed to be active. I'm going to show you for the next half when we get to, they clearly have the double knockout we're referring to. They clearly have a decrease in muscle mass, and that results in exercise impairments in terms of when you force them to run and voluntary wheel running. So, they don't love to exercise, that's for sure. And
Okay.
I'll dive into that next. Maybe you could follow up with an additional question if you have it after seeing some of the data.
Great, thanks.
Are we okay, Maria, from the chat? Any other questions?
Yep, no, that's it for now.
Okay, so, right. So I just mentioned the second bullet point, that, the real surprise we think is, is kind of severalfold. One, that at least in skeletal muscle, there's this functional compensation. And number two, that in skeletal muscle, if you get rid of all of the AKT, genetically, it's not sufficient to block glucose uptake, nor is it sufficient to cause systemic defects in insulin action. And we see normal GLUT4 translocation. And data that we published, I'm not going to show you, we do not see the canonical activation and phosphorylation of AS160. So this is indeed a separate pathway. And that real point I want to hammer home is that insulin signaling and the relative contribution of AKT to GLUT4 and glucose uptake is very tissue distinct. And we, and now the Katawataki group, has shown a dispensable phenotype for muscle. But in the adipocyte, I want to make sure I'm very clear, is that AKT2 is indeed required in vivo to control glucose uptake, and then that can lead to other effects on insulin resistance in not maybe solely due to glucose uptake, but also affecting lipolysis, lipogenesis, and other fat-specific phenotypes. And the last point I really want to just highlight again is that insulin action or insulin signaling in muscle does not equal adipose tissue. And then as an extension, changes in AKT phosphorylation do not equal insulin resistance. I want to really define insulin resistance as a failure to control glucose metabolism and not a defect in AKT phosphorylation. Okay, those can be decoupled as I just showed you. Okay? Doesn't mean one can't cause the other, but they can certainly be decoupled. All right. Okay. So what is the AKT independent mechanism? I'm going to show you a little bit of data here about what that can be. And we became very interested in the AMPK pathway based on some data that we generated with phosphoproteomics. So we worked with Mark Chedrikowski and Steve Gigi's group at Harvard to say in the muscles that lack AKT, if we do look at the insulin-induced phosphoproteome, what are the changes that are really seen? And if you do this, you see a series of changes. And if you do KEGG analysis, one of the largest increases that we see is in AMPK signaling, which is a bit surprising. is never really thought to be insulin regulated. But thus indeed in the AKT knockout, we see this change in AMPK. And if you look at specific targets of AMPK, including ACC, Raptor, and ULK, you can see this selective increase in the AKT knockout with insulin. Okay. So phosphoproteomics suggests that AKT may be inhibiting the activation of AMPK. And we think it's connected to an energetic problem, right? So here's just looking at ultrastructural analysis of mitochondria in the control or the AKT. And you can see in the AKT knockout, I'm not going to spend a lot of time here, they're disorganized, their perpendicular structure is abnormal, and they seem bloated, and there's abnormal changes in the cristae structure. And then doing high capacity respirometry in the Ouroboros, you can see defects in complex 1 activity, and just looking at nucleotide changes, you can see a reduction in ATP, okay? And then to validate, you can look at signaling of AMPK and follow phosphorylation of AMPK by the threonine 172 site. And again, control with insulin, AMPK is not inactivated. Right? This has been known for a long time. However, in the AKT knockout, we see an increase in basal AMPK phosphorylation, and we see this really surprising insulin responsiveness of AMPK, specifically in the AKT double knockout. Again, consistent with somehow AKT suppressing this pathway. Okay, we know a little bit about AMPK in terms of glucose uptake, because there's been a fair amount of work linking AMPK to potentially exercise and contractions ability to drive glucose uptake. And most famously, exercise is an insulin sensitizer. So unlike diabetes, if you exercise or contract muscle, you can shift the dose response curve to the left. So here's just exercise in the open squares, measuring maximal increase in glucose uptake with various insulin concentrations. And this is what Victor's shown a while back. Again, more insulin sensitivity with exercise. Others have shown that AMPK, this is this heterotrimeric this complex here, AMPK, that can be activated by exercise downstream of changes in nucleotides. And more recent work has shown that if you take a muscle-specific AMPK knockout, exercise them, you can prevent the ability of insulin to have that increase in insulin sensitizing effect, right? So here, just to walk you through this, if you contract an animal and you to exercise the muscle and you stimulate with insulin, you can see this increase in glucose uptake, right? This increase in insulin sensitivity, and then that is lost in an AMPK muscle-specific knockout, right? So this increase in insulin sensitivity is dependent on AMPK under these conditions and has no effect on AKT, and there's no effect on AKT phosphorylation. Okay, so there's some evidence that AMPK can be involved in insulin sensitivity. So we asked the question using genetic epistasis experiments, is AMPK serve as this parallel pathway? So Natasha generated a quadruple knockout of specifically in the skeletal muscle by crossing AKT 1-2 floxed alleles with AMPK alpha 1 and alpha 2 to generate this knockout. Here you can see the AMPK alpha isoforms are deleted, and we no longer have AKT phosphorylation. And just on normal chow, these aren't high-fat diet studies, you can see now this quadruple knockout, this inducible knockout, has glucose intolerance, pretty dramatic insulin resistance, and excuse me, and a failure of insulin to stimulate glucose uptake in ex vivo preps of soleus or EDO, right? So this is completely blocks insulin's ability to stimulate glucose uptake in this model. Working with Louise Lantier and Dave Wasserman at Vanderbilt, we did some clamp studies, and consistent with the ex vivo data I just showed, the whole body infusion rate is lower in the quadruple knockout, total glucose disposal is reduced, and the tissue-specific insulin stimulate glucose uptake is reduced in these quadruple knockouts. So collectively, these data demonstrate that deletion of both AKT and AMPK is required to drive systemic insulin resistance in mice under normal chow conditions. So I just want to put forward, let's leave this here. We have a lot more work to do to figure out a lot of these arrows, but this is sort of the classic model that we used to think about for skeletal muscle, that insulin through AKT drives GLUT4, and that contraction through a completely parallel pathway can activate AMPK to drive glucose uptake. This revised model, I think it's just a little bit more complicated. There's a lot more crosstalk that was initially appreciated, in that there's this communication between AKT and AMPK that's involved in the regulation of glucose metabolism. But again, I want to emphasize this, we think is unique to the skeletal muscle, because in fat tissue and primary adipocytes, we have no evidence that there's an existence of a different pathway that can compensate when AKT is deleted. So that is indeed AKT is both, it seems to be required and sufficient to drive GLUT4 and glucose uptake in adipose tissue. Okay, I just want to, before I segue to the growth phenotype, I just want to share a little bit of data that this was all about glucose uptake, but the further question, the next question could be, once the glucose is in the muscle, does AKT really orchestrate where it goes and how it's utilized? So a talented, now dual degree MD, PhD graduate, Anna, worked with Tara Tesla, initially in Josh Rabinowitz's lab, now with her own lab at UCLA, to use stable isotope labeling, here C13 glucose infusions, to infuse into the skeletal muscle, to infuse in vivo into either control or skeletal muscle AKT knockouts with and without insulin, to ask the question, does AKT control more distal steps in glucose metabolism beyond just glucose uptake, as I just walked through for the last 15, 20 minutes. So the short answer is indeed it does. We see this really interesting effect at the level of glucose 6-phosphate. So I'm just walking you through this. This is a data presented as the labeled fraction normalized to serum labeling. So this is C13 glucose infusions with insulin injection. And you can see in control, you can see this increase in labeling of glucose 6-phosphate. And this glucose 6-phosphate, this increased fractional labeling, is lost in the AKT knockout. And pretty much everything else downstream, including unit peak glucose and glycogen synthesis, right? So this, again, this is the label, this amount of glucose label coming from what we infused into the glycogen pool. Insulin increases that in control, and the AKT knockout is lost. So just step back and just say, you know, we think that AKT can be dispensable for glucose uptake, but it does indeed seem to be important for glucose utilization. Exact mechanistically unclear. You can certainly suggest a role for hexokinase, which may be in play, but I just want to be cautious in the way I describe these data. You could get the same answer in terms of decreased labeling in the G6P pool in the AKT knockout if you have increased unlabeled contribution from glycogen that was already stored. So if there's an increase in glycogen breakdown that was not suppressed by AKT, you could still get this same answer on labeling. So at this point, we really just know there's a change in the glucose 6-phosphate pool in terms of labeled fraction, but really mechanistically, is it hexokinase? Is it a glycogen contribution? I can't yet say. Okay, I'm going to skip this for the sake of time, but basically everything downstream of G6P kind of tracks with decrease in labeling. And you can see this here in the distal glycolytic intermediates in terms of the fractional labeling, and also in TCA intermediates. There is some increase, but the absolute contribution compared to a control is reduced, right? So there's decreased glucose contribution to the TCA cycle. All right, so just stepping kind of back, I think this is we're going to be revisiting the classic model that AMPK controls GLUT4 in an independent, distinct pathway that AKT does in terms of insulin. But based on the genetic data that we have, and also from other groups like Katawaki, I want to suggest that there's really a new model of how AKT is involved in glucose metabolism in the muscle. It seems to prevent this energetic pathway involved in AMPK, and that once you remove AKT, this pathway can serve as an important regulator of GLUT4. But there's, I think, this underappreciated role for AKT in glucose utilization, in addition to GLUT4, that we're really interested in exploring in the future. And in the last part, I'm going to show you a little bit about mitochondrial biogenesis. So maybe I'll just pause for a minute if anybody has any questions about glucose metabolism, because we're going to shift gears and talk for the last 10 minutes, 10, 15 minutes about AKT's role as an anabolic signal for muscle hypertrophy.
Oh, we did have a question in the chat.
Okay.
AKT is reported to inhibit AMPK by serine 485 to 492 phosphorylation. Have you examined AMPK serine phosphorylation in your model?
Yes, we have. That was the first place we looked. We do see that in cells. If you treat with insulin, we see changes in that phosphorylation site. And then if you use a small molecule AKT inhibitor, you can block that. However, in vivo, when we knock out AKT, we see no changes in that phosphorylation site on AMPK. So either there's other kinases that can compensate in vivo, or there's another mechanism in play. So thanks for that question. That was certainly one of the first places that Natasha looked for this regulation. Okay. All right. For the last 10 minutes, I want to talk about maybe the less surprising finding, And I already kind of alluded to is that when you get rid of both AKT isoforms, you can see a significant reduction in the muscle mass. And what we really wanted to understand is what were the downstream signals for that, and are there functional consequences? And this is work that was been previously published about 15 years ago or so. Really, two different groups have shown the activation of AKT is sufficient to drive muscle hypertrophy. This is, I think, Walsh's group. David Glass has shown something similar. If you make a transgenic AKT skeletal muscle overexpressor, you can see an increase in the muscling of the animal, and then there's an increase in the cross-sectional area here, staying either the tibialis or the MG here. So as you just see this bigger fiber size quantified here on the right. So clearly activation of AKT is a potent anabolic stimulus in animals. And what we really wanted to understand is what was the mechanisms mediating that, and exploring really the distal control and showing if it's required for growth. Okay. So we're going to really ask these two kind of questions. Okay, thinking about IGF signaling, IGF, like insulin, activates AKT. IGF is obviously a pro-growth signal. If you get rid of both AKT isoforms, you can prevent IGF's ability to phosphorylate and inhibit FOXO and also activate mTOR. Deletion of both AKT isoforms, just showing you a little bit more data related to the muscle phenotype. There's a significant reduction in all fiber depots that we measured, tibialis, EDL, soleus, gastrocnemius. The opposite of the previous slide is if you get rid of AKT and you measure the cross-sectional area, you see a shift to the left. So they are smaller fibers, again, consistent with less hypertrophy in these animals. And related to that question I answered earlier, the defect in muscle mass is also associated with significant weakness of the animal. So there's just grip strength is reduced. They really don't love to run. If you just do voluntary wheel running, these animals are quite resistant to that. And if you force them to run on doing maximal distance run or time to exhaustion on forced treadmill test, you can see that there's a significant decrease in exercise performance in animals that lack AKT in the skeletal muscle. To isolate these effects, you can look at the, you can do contraction assays in ex vivo tissue. This is in collaboration with Teji Khurana at Penn Department of Physiology. And you can just measure force curves, and the absolute force curve and the AKT knockouts in the red is reduced in both the fast twitch and slow twitch muscle. Not so surprising because the muscles are smaller. If you normalize then to the cross-sectional area and measure really specific force, it seems to normalize the EDL phenotype. However, the soleus still seems to be weaker. Okay, so there seems to be that it's largely dependent on the size. It corrects most of it, but there is some inherent problem in the slow twitch muscle in terms of muscle strength and performance. And we think that's related to the oxidative phenotype, which I'll get to in a second. Okay, mechanistically, the AKT knockout, it has lower muscle, we think smaller muscles because of a defect in protein synthesis. And here is just showing deuterated water labeling experiments to measure the fractional synthesis rate in these animals. You can see a 25 to 30% reduction in D2O incorporation. We don't have great assays to measure protein breakdown. We didn't see any changes in the canonical attrogenes, which was a bit surprising, but indeed there's no gene expression changes in MERF and attrogene 1. Measuring proteolysis ex vivo, tyrosine release, we didn't see any real change. If anything, proteolysis and tyrosine release was lower, certainly not higher. And there was no changes in steady state, either plasma or amino acids in muscle that would be consistent with breakdown. So maybe an increase in amino acids. So we think largely the muscle mass phenotype is driven by a defect in protein synthesis, not an increase in protein breakdown. Okay. I promised to talk about mechanisms downstream. So it's been put forward by Glass and others that mTOR and FOXO may serve downstream of AKT to control muscle growth. But again, there's never been in vivo evidence that directly linked these pathways and asked the question, which ones are required and sufficient to control muscle growth downstream of AKT. So we tested this. We did these experiments in vivo to test the hypothesis and really define which ones were driving the phenotype. So we generated a series of triple knockouts. Here we knocked out FOXO, downstream of AKT, so you have an AKT FOXO triple knockout, or we activated TOR by inhibiting its negative regulator TSC. So we generated an AKT double knockout plus TSC to generate a triple knockout. Okay, here's the Western blots. And doing either of these alone, either knocking out FOXO or knocking out TSC1 on top, did not correct the phenotype of the muscle mass. Okay, so no changes really in growth. However, what Natasha did is she generated a combination. She asked the question, well, maybe both are required, but neither is sufficient. So she generated a AKT, TSC, and FOXO quadruple knockout. Okay, let's just demonstrate. Here's a Q knockout. So another quadruple knockout. This one's distinct from the glucose part of the talk. This is a different quadruple knockout here. Again, the idea is to activate TOR and inhibit FOXO, these two competing pathways in the absence of AKT, and ask the question, are they both required and sufficient for growth? And the phenotype is quite striking. It completely normalizes the body weight phenotype. If anything, the muscles are larger, not smaller, and they appear redder in color. So we think maybe there's a fiber type, which I'll show you in a second. And unlike the AKT knockout alone, which had smaller cross-sectional area, these shift to the right, and these are much larger fibers. So you have really unrestrained muscle hypertrophy when you activate TOR and inhibit FOXO. But again, emphasize neither alone does this. And this normalizes protein synthesis. I'm going to go quick here for sake of time, but it also normalizes exercise performance, at least at the whole body level. And lastly, I just want to kind of focus on last point, that AKT seems to control the oxidative fiber type. And here I'm just showing you the soleus and the EDL standing for different myosin heavy chains, just quantified here on the right. So in the soleus, the AKT knockout reduces the slow, canonical slow myosin heavy chain, so MHC1, increases MHC2A, and that's normalized in the quadruple knockout. So AKT controls the oxidative phenotype and the oxidative fiber type of the muscle, and that's dependent on these two pathways. Consistent with that, you can see a reduction in whole cell mitochondrial complexes. I'm going a little bit fast here for the sake of time, but just want to maybe spend a little bit more time here. Again, this is doing Ouroboros to measure mitochondrial respiration. Again, as I already showed you before, the AKT knockout has reduced O2 respiration at state 2, state 3. That is, again, corrected with the quadruple knockout. It's completely normalized. Okay. We'll skip that for the sake of time. The last 30 seconds is, why should we care, and what's the physiological relevance of this genetic model? We turn to a pretty relatively straightforward assay where you can do a casting or immobilization-induced muscle atrophy, where you take a hind limb of an animal, you can cast that leg. That leads to actually significant reduction in soleus and gastrocnemius weights within 5 to 7 days, which has been reported to be associated with decrease in AKT phosphorylation and mTOR phosphorylation. So we took advantage of this model, casted our own animals. Again, we recapitulated that the soleus and the gastrocs are lower than it's associated with this modest impairment in AKT signaling, and asked the question, if you activate two pathways that we just defined, they're required and sufficient to control muscle growth and the oxidative phenotype, are they sufficient to normalize casting-induced muscle atrophy, even when AKT is not perturbed genetically? So here is a double knockout here. There's a different double knockout. Sorry for all of the different knockouts, but hopefully I'm clear here. This is a FOXO TSC double knockout, inducible, which is a tamoxifen-inducible skeletal muscle specific version. And you can cast these animals, and you can see here in the control, you see about 30% reduction in muscle mass in casting, in the soleus, and about 20% in gastrocnemius. If you do this in an FOXO TSC double knockout, you can significantly reduce the amount of muscle loss that occurs in casting. Now it's only roughly 10% and roughly 5% in the gastrocnemius. And again, we think that's due to the protein synthesis. And this is just the sunset method measuring puromycin incorporation. And you can see in general, the double knockout here has much higher labeling with puromycin incorporation, really consistent with an increase in protein synthesis. Okay, so to conclude, mice lacking both AKT isoforms and muscle have reduction in mass. It also seems to control the oxidative property of the muscle, and I have data that I showed you about mitochondria and also about the fiber type. These reductions in mass are also associated with impairments in muscle strength and exercise performance. And then using genetic epistasis experiments, we show that AKT signaling through both mTOR and FOXO, but neither alone, are both required and sufficient to drive muscle mass, mitochondrial function, and exercise performance. And this, we think, is relevant, at least in the case of immobilization-induced muscle atrophy. This is what I just mentioned to you. This is the pathway. I just want to acknowledge the people that really contributed to this work, specifically Natasha Joswal, who was a postdoc fellow with me for the last several years, that really spearheaded all of the muscle AKT work. She's now launching her own independent lab at Purdue University in January. Also like to specifically thank the funding, including the NIDDK, and be part of this AMP consortium that really helps us understand genetics of type 2 diabetes and the relative contribution of muscle, liver, and adipose tissue to those phenotypes. So with that, I can take any questions for the last couple of minutes. Happy to do so. Thank you.
Thank you so much, Paul. We don't have any other questions in the chat right now, but if anyone has questions, just feel free to unmute yourself and ask them.
I had a quick question, Thomas here. So great talk, Paul. I was wondering if you have deleted these specific isoforms in the heart or only in the skeletal muscle.
We haven't spent, thank you for the question, Thomas. I tell the reason I harped on the HSA-CRE model approach we did is we had previous experience using a non-cardiac excluded CRE, which hit both heart and muscle. And I tell you, if you do that approach, there's not many animals that make it past weaning. So we predict that there's some issues in terms of a cardiac phenotype, but we haven't done anything more related to that. But yeah.
That's okay. Yeah, I was just curious, yeah, how it pans out in another muscle subtype. That's what I was more curious about.
Yeah, I suggest that there's a significant cardiomyopathy that's going to happen, and I think the best way to do it would be in an inducible system and really have it be just cardiac focused. But that's the anecdote that we kind of did it, but we have no real data other than what I just kind of told you.
Sure. One last thing I want to just mention, and as these animals are aging over time, have you looked at their immune cells around the muscle, how they develop, how they play a role in muscle development, and would they have any effect on the phenotype itself of glucose metabolism or hypertrophy?
We haven't aged animals for quite a while. I do want to emphasize, and most of this work we have published recently. We've done a series of additional experiments where we do the inducible knockout to really isolate acute effects. So we have the congenital, but we also do the tamoxifen inducible system. We have had a couple cohorts that went longer, 6 to 8 months, but certainly not aged phenotypes. And there's definitely a large influx of inflammatory pathways that are where you do an RNA-seq sort of experiment. We haven't decoupled how and what's their relative contribution to the glucose phenotype, because frankly, in the AKT knockout under normal conditions, there's not much of a glucose phenotype. But there's certainly a large influx of inflammatory cells in the muscles that lack AKT. Now, is that secondary to the atrophy that we see or the failure of muscle growth? That I don't know. We don't see them early if we do these tamoxifen inducible systems. We certainly can see muscle loss before an inflammatory component. And the last point I would just mention is Katawaki in his Nature Communications paper with different muscle AKT knockouts, he did more of aging phenotypes and definitely demonstrated with age there's effects in the bone and other sort of secondary effects on insulin action or insulin resistance, et cetera. Hopefully that answers your question.
Thank you so much, Paul. That was great. Thank you.
Yeah, thank you for showing up, and thanks for the questions. Alan, go ahead. I see your hand again.
So the question is, as you know, adipose tissue is the most insulin sensitive tissue in the body. I don't know if there's any other one that's more sensitive. But I wonder if you've discovered the reason. Maybe it's because of AKT2 playing that very, very special role in adipose tissue. Do you think that's the case?
I think that's a very provocative hypothesis that we're very interested in. We don't have an answer for why AKT deficiency in fat tissue causes this profound phenotype. It causes the reduction in signaling that we see, and then also causes this requirement for GLUT4.
Does it also affect the ability of insulin to stimulate lipogenesis in adipose?
Yeah, and lipolysis. Sorry, I should have just kept going. And we have a failure of lipolysis. We don't see lipogenesis in the fat cell. Everything that you would canonically think about what AKT does in terms of glucose and lipid metabolism is indeed true in the adipose tissue, at least in when we do the AKT2 fat specific knockout. So is there truly substrate specificity in the fat versus the muscle for these different isoforms? Is it really just levels and that there is no functional compensation that can happen? You know, I don't have a good molecular explanation for that, but I like you, I think it's clear that the role of this kinase in fat tissue is really important for both adipose function and then subsequently the systemic control of liver, muscle, and really the whole organismal level phenotype. I don't have a great answer for why. You know, we're doing sort of more detailed phosphoproteomic analysis in the different tissues to see if there's different substrates that may be in play. But a lot more work to do, Alan. So I appreciate the question.
Great seminar. Thank you.
Thank you. Yeah, please. See a hand up.
Yeah, hi Paul. That was fantastic. This is Kutaiba Kawi from Memorial University of Newfoundland in Canada. I really enjoyed the talk. Two quick questions.
Sure.
You talked about GLUT4. I'm not sure if you looked at other isoforms of GLUT1 or the others.
Yeah. Yeah, so in the original paper that we put forward on the glucose phenotype, we did look at GLUT1. I will be honest with you, we just looked at total levels of GLUT1, and we did a very crude biochemical fractionation of GLUT1 and GLUT4 in that paper. That's why I tried to showcase some of Natasha's newer work in single fibers to actually measure GLUT4 translocation. As you may or may not know, I'm not sure if you do these studies yourself, GLUT4 translocation is incredibly challenging to do in vivo. I'm coming from, being trained in Maury Birnbaum's lab, who spent a lot of time thinking about that. So we like this trick with what David has used in the past. So that's all I can say. The general expression of GLUT1 was the same. Yeah, that's where I'll leave it. And the second question?
Fantastic. So second question, have you tried to fast those AKT knockout mice and see what happens to circulating fat and ketone body production after fasting in those animals?
We did some fasting, actually more focused on trying to see if there was atrogene changes. We don't, or at least on the normal chow. Now the high fat diet is different, and we're kind of doing those studies now. At least on the normal chow, we did not see big changes in fasting glycerol or fatty acid levels in fasting and refeeding. But I will tell you, it kind of relates to Alan's question. That is not true when you manipulate AKT signaling in the fat. You cause really significant changes in circulating fatty acids, glycerol as markers of lipolysis, reesterification, et cetera. So, yeah, I just want to emphasize, we really do think skeletal muscle and fat, in terms of the canonical insulin signaling GLUT4 pathway, they're more nuanced and they're more tissue dependent, that we got a lot more work to do to really parse out the mechanisms.
Yeah, I agree with you. Fascinating studies. Thank you, Paul.
Thank you.
Thanks so much. And I see we're at time, so I guess we should stop. But this has been great, and we will we've been recording it, and we'll post the recording so even more people can enjoy the seminar.
Fantastic. Thank you, Maria. Thank you, Mackenzie. Thanks everyone for the opportunity and for the questions and for attending today. Appreciate it.
Thank you, Paul.
Thanks all. Bye-bye.